Significance of mitosis. Mitosis is responsible for the growth which we see in multicellular organisms. Mitosis also restores the nucleocytoplasmic ratio, which is responsible for the proper division of a parent cell into two daughter cells, and the most important significance of mitosis is cell repair. Meiosis. Meiosis is also known as reductional cell division, as it helps in restoring and maintaining haploid phase of cell division cycle, because sexually reproducing organisms, when they fertilize, they form diploid cells. Hence, meiosis takes place in them so that the haploid phase is restored. The gametes are haploid in nature, and when this sexually reproducing organisms, they'll mate or fertilization will take place, cells will again become diploid in nature. Meiosis is divided into two phases, meiosis 1 and meiosis 2. In meiosis, pairing of homologous chromosomes as well as their recombination takes place. Meiosis 1. Meiosis 1 is marked by prophase 1, metaphase 1, anaphase 1 and telophase 1. Prophase 1 is further marked by 5 stages: leptotene, zygotene, pecitine, diplotene and dikinases. You can see in the diagram prophase, then metaphase 1, anaphase 1 and telophase 1. Leptotene is the stage where chromosomes becomes visible. Now we can see chromosomes under microscope. Zygotene is that stage in which the association and pairing of chromosomes known as synapses takes place, and this synapses, it is formed by forming a unique structure known as synaptonemal complex. Then is pecitine. Leptotene and zygotene are short-lived stages, and pecitine and later stages are longer in comparison to these two earlier stages. Pecitine is marked by presence of recombination nodules. In case of diplotene, the synaptonemal complex, it starts to disappear and a X-shaped structure known as chiasmata is formed, which involves the separation of these associated or paired chromatids or chromosomes. Next stage is dikinases. In this stage, meiotic spindle starts to form and it leads to further separation, initiation of separation of these chromatids. Metaphase 1. The bivalent chromosomes so formed, they align on equatorial plate. You can see it in the diagram. Anaphase 1. In this phase, homologous chromosomes, they separate, whereas sister chromosomes, they remain associated and attached to their centromeres. Telophase 1. In telophase 1, nuclear membrane, nucleolus appears. It starts, this nuclear membrane and nucleolus starts to form, and further cytokinesis occur, and hence you can see after telophase 1, two cells are present.